Good afternoon, everyone. I'd like to thank EBRAINS basically for having Nicola and myself presenting not EBRAINS, but MEBRAINS, a multi-level macaque brain atlas. Nicola, so this is a brain atlas. Why is it important to create a new macaque template if our ultimate goal is basically to understand and, if possible, repair a broken human brain?
Well, Wim, creating a macaque atlas is important because for human brain research, because macaques combine a close evolutionary similarity with humans, with the fact that we are able to perform invasive experiments with them, which can't be carried out with human subjects. So, for example, with macaque monkeys, we can measure the spiking activity of individual neurons or of small groups of neurons while the monkey is performing tasks involving, for example, memory or movement. We can also use optogenetics, which is a really powerful method because it enables us to go a step further and turn specific neurons or pathways off. So then you can see which one of the pathways is actually necessary for a brain function, or if it's sufficient to enable that brain function. With macaques, of course, you can also do histology, which you can do with humans, but with macaques, with animal models in general, this has the advantage that you can control the perimortal situation much more than well, you can control it, which you can't in humans. Okay, and this means that you get an experimentally more gold standard data and reference for interpreting human brain data. Macaque monkeys can also be used, for example, for lesion studies to ask what changes happen in the brain when, for example, a specific brain vessel is damaged. You can also perform track tracing studies aiming to identify the pathways which connect different brain regions. So all of these experiments are things that you would not be able to do in the human brain, but they are essential to shed light on structure, function, organization principles.
Okay, Nicola, so this is quite theoretical and very interesting, but can you explain a bit more specifically what kind of information you wish to anchor to this macaque template and how it basically makes this model more useful for the user?
Right. Well, as you can see in the slide, what we want to do is create an atlas which integrates data from different spatial and temporal scales. So along the spatial scale, we would want to be able to have data, ranging from the microscopic level. So for example, single cells, through local circuit organization, cortical layers, connectivity between areas. And then we want to be able to also move up into the centimeter and several centimeter scale, where we would then be able to look at, entire brain regions, so-called macroanatomic landmarks, or gyri and sulci. And then, of course, along the time scale, we don't just want to look at ex vivo data, so post-mortem data. We also want to be able to look at things that are happening during different time scales. So across the lifespan, but also just within some minutes or even milliseconds, changes in neuronal activity. So all of this has to be combined into a single atlas, which would enable simultaneous analysis of all these kinds of data.
In summary, you want to anchor multimodal, multi-scale information in the same space, in the same three-dimensional space. But we know that these kind of spaces, atlases already do exist. So why going through the effort to build a new template, a new atlas, Nicola?
Well, yes, you're right. Such atlases already exist, and actually, macaque monkey atlases have been around since the 1960s. The original atlases were all just two-dimensional. They were basically books, okay? But even the modern atlases, the ones that are now three-dimensional atlases existing, they show limitations because they're based either on a single subject, that is the example of the D99 atlas, or so they don't reveal structural variation across individuals. And other atlases, such as the NMT template, they have been used creating only one modality. And although then you can see things to do with inter-individual variability, so variability between subjects, you only have the one modality. And since last century, we also know that multimodality is important to understand how the brain works, because no single modality is sufficient to explain brain function. So then what we wanted to do was create a template that integrated the positive sides of existing atlases and tried to overcome the minus points. And I know that you were heavily involved in this, Wim. Well, you led this process. So tell us a bit, please, about how the MEBRAINS template was created.
Yeah, well, actually, you're completely right, Nicola, that we wanted to go beyond what was existing. And as in the previous slides, you could see in the previous slide, these templates look actually already very, very nice. But we specifically wanted to have multimodal information and from many subjects in one, integrated in one template. And what we did is we acquired images, T1 weighted and T2 weighted MR images from single subjects, specifically 10 subjects, or we acquired actually many more subjects, but in the present version of the MEBRAINS Atlas, we only included 10 subjects. And then we used all kinds of algorithms to merge these images with their individual variability. Of course, it's not an easy task. It was even challenging to merge them into one single template. And we tried many different algorithms and landed upon one particular one which performed exceedingly exquisitely well. So this is an example of the T1, sort of like the MEBRAINS T1 image. This is of the T2 weighted image. And we also had the luxury to acquire CT images. And we did that in 9 of the 10 animals that were subject in the MEBRAINS, in the current MEBRAINS template. Once we had that, once we created that, we also recreated basically the cortical surface in 3 dimensions, as you can see here on the right-hand side. For the left hemisphere, where the back of the brain is on the right and the front of the brain is on the left. And then you can artificially inflate the image until you get sort of like what is called with jargon a folded image, which has the advantage that whatever is buried in a sulcus here in reality starts to appear on the surface of this folded image. And then when you cut, you can even go further and show the entire cortex, in this case, in one what is called flattened image of MEBRAINS.
Wow, this is really nice, Wim, and especially the flat map isn't, apart from the fact that the resolution of this template is as good as or even better than the existing ones, the flat map is an additional bonus point. But now tell me something, now that we've got a nice template to which data can be anchored, what can researchers actually do with it, and what kind of information can be added to this atlas then to make it useful?
Well, Nicola, as I said, it was challenging to create this, actually much more challenging than I naively thought when we started with this enterprise. But we had in mind that we had already quite a lot of data available that we wanted to anchor to the template to make it usable and to make it interesting to use, basically. And one of the things, one of the obvious functions, basically, that we wanted to anchor is retinopathy. And it's already known for more than a century, or since more than a century, basically, that the brain, the cortical area, but also the cortex contains maps of the visual world. These are called, or from the retina. For example, Holmes, more than 100 years ago, he deduced from patients with brain lesions, very focal brain lesions, mainly or typically from soldiers which had wounds, shot wounds in their head, but which survived, obviously, and which had very restricted lesions in their visual cortex. Visual cortex in the human located in the back of the brain, in the occipital lobe of the brain. And here you can see an image of a medial wall of the occipital lobe with a large sulcus, the calcarine sulcus, and that's where the primary visual cortex is located. And what Holmes found basically is when patients had lesions in the upper part of the calcarine sulcus, they had a deficit in the contralateral lower quadrant. When they had lesions in the more ventral part of this calcarine sulcus, they had deficits. They didn't see anything. They had a blind spot in the upper contralateral quadrant. Moreover, when the lesions were restricted to more posterior portions of the calcarine sulcus, they were specifically impaired when stimuli were presented near the fovea, near the central part of the retina. So this basically showed very nicely that there is a map of the visual world in the visual cortex. So basically the visual world is represented through topographically organized maps. It's not just one map, but multiple maps. We even don't know at this very moment how many maps there are, probably 30 plus types maps. And these retinotopic maps basically correspond to visual areas. The nice thing about this is if you can then map these maps, if you can identify these maps, you also know in vivo the boundaries of these visual areas. So this is in the human, and people use that actually to their advantage already since the beginning, or since the last decade of previous century, 95, I think the first papers showed up in the human literature where they started mapping out the visual cortex in detail in vivo in humans. Lucky enough, in the macaques, we knew that already from electrophysiological recordings, from tracer studies, and so on and so on. The macaques have sort of like a very similar functional organization as the human. But unlike the experiments that were done in the 90s of last century in humans, we only started to do these kind of topic mapping experiments in monkeys after.
Oh, but tell me something then. How did you acquire this data in the macaque monkeys, and how did you integrate it into MEBRAINS? So, you know, because you just said that these maps have already been known in the past.
Yeah. So this is easy to say. It was not that easy to do. And what we basically did is we trained monkeys to fixate here to my laser pointer, basically to fixate very nicely to a specific point in the visual field. And then we presented all types of stimuli on a screen. And we put these monkeys in a scanner. They were awake and they were sort of like rewarded just to fixate to the center of the screen. And then in one type of stimuli, here these wedges, they were shown in the background and they slowly rotated either clock or counterclockwise around that fixation point. In another set of experiments, basically, or runs acquired in the same day, we presented annuli or rings that were initially small and that slowly grew until they reached peripheral parts of the visual field. So we had expanding and contracting rings. And as you can see here, the carriers, sort of like what filled the wedges and these annuli, contain different types of stimuli. Here, these were moving monkey faces. Here, there were walking human bodies, but we used all kinds of stimuli, complex objects, checkerboard stimuli, and so forth, while these monkeys were fixating. And we did that in quite a large number of macaques. And I think that's pretty unique. I don't think many labs do have the capability to acquire this kind of written topic data in many, many subjects, macaque subjects in this case. 4 of them were female monkeys. When you do these experiments, well, we mapped the eccentricity. This was a little bit of a caveat. And this was also dependent on the scanner, basically. We could not initially acquire larger data. We only mapped up to 12.5 degrees eccentricity. We mapped polar angles. And then when you have that information, when you have eccentricity and polar angle information, you can calculate field sign maps. And this will become clear in the next slide. When you analyze that data, we were able to locate in every individual about 19 written topically defined areas, which is quite unique, because then you can combine that information and you can create what is called a probabilistic written topic atlas. So all the monkeys did the same experiment, all the data were analyzed in the same way, and then you combine this data, and then the beauty of EBRAINS is that you can project that into MEBRAINS. We did that by registering surface-to-surface information from the individual surfaces of the subjects to the surface of the MEBRAINS, which I showed in one of the previous slides. How does this look like? Well, here you see this brain, this artificially recreated cortex of a single subject on which we painted the activity, the eccentricity activity, where we artificially color-coded regions that respond, as you can see here at the pointer, to foveal locations in purple, dark blue color. And when you gradually go to more peripheral locations of the visual field, up to 12.5 degrees in this case, it becomes green, yellow, red. And that's exactly what you see here painted upon the cortex of this right hemisphere, where the back of the brain is here, front of the brain is here, and this is a temporal lobe. And you see foveal regions or voxels, or in fact neurons that are located there, responding to foveal parts of the stimulus are color-coded in blue. And gradually you get larger eccentricity representations when you move here from this part of V1 to more medial parts of V1. This is painted upon the brain itself. Then you can do the trick to, as I explained previously, you can project that information, that functional information, to the gray-white matter boundary, the same information, but at a different level of the cortex, where the cortex transitions to the white matter, and then you can blow it up with a nice advantage. Here, things are buried in sulci, as you can see. When you start blowing it up, when you make the folded brain, you see that everything starts to appear on the surface. Everything becomes visible in one single image. Then if you cut, then you can do the complete flattening. Right now, there is stuff hidden behind what you're seeing here and the medial wall of this right hemisphere, and you get a fully flattened hemisphere. So this is how we do that in a single subject. So this is a written topic map, center representations or foveal representations in blue, more peripheral, going from green, yellow to red. This is one brain. And obviously, you can do the same thing for these polar angle experiments, where you measure which part of the cortex is responding to a situation when the stimulus is in a particular location in the polar angle. For example, here in green, the upper vertical meridian. And as predicted from that Helmut slide, indeed, ventrally in visual cortex, you see these green bands. So here are the vertices, in fact, neurons that are responding when the stimulus is covering the upper vertical meridian. And then you have in between blue regions that are responding to the contralateral horizontal meridian. And then you have red regions in the dorsal part of visual cortex that are responding to situations or times that the stimulus is in the lower vertical meridian. And you see this alternation from horizontal meridian to vertical meridian, horizontal meridian, vertical meridian, both dorsally and ventrally in the visual cortex. Yeah. This information, this combined information, it basically enables us to in vivo delineate the areas, the consecutive areas, area V1, V2, V3, V4, and so forth, and so forth. Again, this is from one individual. This is another individual. And then you can calculate the field sign maps. And basically, it's a very simple concept. In some areas, you have what is called a mirror map from the visual world. So the map in the brain is a mirror from the visual world. In other words, when you move from left to right in the visual world, you will move in the brain from right to left. For in non-mirror maps, when you have a non-mirror map, if you move from left to right in the visual world, you move also from left to right in the brain. And that's color-coded here in blue and in red. You have the mirror maps in blue and the non-mirror maps in red, which indicate where these borders between consecutive areas are. Again, this is one subject, another subject, and you do this for all these 13 subjects that contributed to our probabilistic topic atlas. Then you can average. That's sort of like a poor man's solution. And you see this is the average of all the data that we get. And it becomes a bit blurry. Obviously, it becomes blurry because there is anatomical and functional variability between different animals. This is the average eccentricity map for the left hemisphere and the right hemisphere. The average polar angle map, and you see these transitions between horizontal and vertical meridian representation becomes a bit blurry simply because you're averaging. And then you can sort of like deduce the aerial boundaries which are here indicated in white. But this is already a good start, right? And we provide that also in MEBRAINS. A smarter thing which preserves, or a smarter thing to do, which preserves sort of like the intersubject differences while still allowing generalizability, is to create what we call probabilistic maps. And here, for example, the outlines, the different outlines that you're seeing here on this flattened left hemisphere of MEBRAINS, basically, are the V1s that we map in every single subject. So every single subject data is there. And then you can say, okay, which voxel, which part of V1 basically belongs with a high probability to V1. And you can arbitrarily say, okay, in 50% of the cases, I want to have it belonging to a particular area, in this case, V1, and you color code it. And then you get sort of like a probabilistic estimate of V1. The nice thing here is that when you have a new brain where you don't map the retinopathy and you register the anatomy based upon T1 or T2 weighted images of that new individual, then you can sort of like estimate with pretty good confidence that the voxels of that subject here that are color-coded yellow here in MEBRAIN will also belong to V1 in that new subject. So basically, you do not have to map anymore all these written top, or you don't have to perform these written topic mappings in these individual brains. Of course, you can do that in many areas. This is, for example, V4. You see V4, these are the individual outlines of all the subjects for V4. It becomes more variable. So the yellow range, the 50% probability estimate of V4 in MEBRAINS becomes a bit smaller than what you saw in V4, in V1. This is for PITD. This is an area slightly more ventral towards the temporal pole of infratemporal cortex. And you see, again, more variability. The more you move away from V1, the more variability you will get between different subjects. Anyway, once you have all that information, you can put that on a brain. This should move. And you can sort of like paint the 50% probability boundaries of each area on, in this case, the MEBRAINS template. And again, when you have a new monkey, you can register that anatomy to this brain, and then you have sort of like a pretty good chance that if you stick in electrodes or injection needles or whatever you want to do in this animal in this particular location, that it has a 50% probability that it belongs to one of these areas. So this is a way to map in vivo visual areas, but this is not everything we can do to understand functions of the brain, of the visual cortex in this case. So Nicola, what else can you actually add to our MEBRAINS template?
Well, you mentioned that you've got very many areas involved in the processing of visual stimuli, and these areas have to talk to each other somehow, and they do so via fiber tracts, okay? And not just visual areas for the visual system, but in general terms, areas must all communicate with each other to be able to enable function, okay? So MEBRAINS also contains information, data describing the distribution and density of fiber tracts, okay? And there are several methods available with which we can study fiber tracts, and each one of these methods provides different kinds of information about brain connectivity. This schematic drawing that you see right now on the slide depicts tractography, which is a modern, non-invasive method, meaning it enables visualization and analysis of white matter pathways in the living brain, okay? And then we can see how different brain regions are structurally connected with each other. And it is based on the fact that water molecules move along the axons, and the direction in which they can move are constrained by, the length of the axon, okay? And scientists can model these diffusion patterns, and thus tractography can enable reconstruction of the probable connections between brain regions, okay? Now, the problem about this method is that it only provides an indirect estimate of the course of axons in the brain, and actually not visualization of the axon itself. This latter aspect is one that has been or is done by means of a classical method which is called tracing technique, in which a tracer substance is injected into the brain. And we would talk about an anterograde tracing map if the tracer is injected into the brain region that is sending axons out, okay? And then this tracer travels along the axons and reaches the target area. So then we can see the output pathways of the region that received the injection of tracer. And then in contrast to anterograde tracing, we've got retrograde tracing, where the tracer is injected exactly in the opposite position. So it is placed in a region which receives projections from another one. It receives axons from another area. And this tracer is taken up by the axon terminals. It's transported along the axon back into the cell body of that cell, and then we can visualize the area that sent those inputs, okay? Now, the macaque monkey is a particularly valuable model because it enables us to look both at in vivo, at tractography studies with the macaque monkey, but you can also do these ex vivo, these anterograde and retrograde tracer studies. So you can compare the results of both methods with each other. And this is particularly important because the tracing studies, so anterograde or retrograde tracing studies, they're considered to be the gold standard. So then whenever we look at tractography studies in humans, we can infer things based on what we've learned from comparing tractography and tracing data in the macaque monkey. Wim, but I know also you didn't just do retinotopy, you've also been involved in tracing studies. So can you tell us something about what people would find in MEBRAINS?
Yeah, that's correct, Nicola. And basically, in the particular study, we used this technology, anterograde tracing, and specifically, we injected an area in the ventral midbrain of macaques, the ventral tegmental area, which is known to house quite a lot of dopaminergic neurons. This was done in collaboration with Natasha Issa from Japan. And basically, these anterograde tracer injections revealed in high detail the anterograde projection pattern, sort of like where the axon terminals originating in ventral tegmental area, specifically in the dopaminergic neurons of the ventral tegmental area, ended up to. So this is one example where this highly precisely tractography information can be implemented in the template. So this is a classical tractography approach with the advantage of being highly specific to target sites, Nicola. An important limitation is that these animals need to be sacrificed and that the connectivity information comes only from a few sites that you can inject in any given animal, right? So these are basically costly, very tedious experiments, and limited information per single subject can be gleaned. So what's a more efficient way to study connectivity?
Well, you'd want to be able to do whole brain analysis, for example, and you wouldn't want to have to sacrifice the animal at the end of it, right? So that's why this kind of data here, which is provided by the MADI 700 data set, is so unbelievably valuable. Okay, it's because we've got data acquired in vivo, and it has already been processed so that users can do their own tractography analysis. So this data set here provides data on the course of axons throughout the entire brain at an isotopic resolution of 0.7 millimeters, so quite a high resolution. And then you could, for example, look and see, well, where would, the axons that I saw now with my ex vivo experiment, you can see that you would also be able to visualize them here. But then besides the VTA axons, you could also look, for example, at axons of the substantia nigra, which also innervates or provides the brain with dopaminergic innervation. So you can look at different other systems and not just one single one.
Very cool. Also very cool colors, of course. But we talked about function and we talked about connectivity. So you can talk about networks and things like that. But what other type of information would be important finally to understand how the brain functions, and information that we can sort of like implement in the MEBRAINS template, Nicola?
Well, that's a very good question, Wim, and the answer to that would basically be receptor maps. Okay, so what you see here is just a drawing of two neurons, and you've got this long axon here, and information flows within a neuron from the cell body along its axon, and then it reaches this point here, which is basically almost like a dead-end street, okay? And it's the point where this neuron establishes a highly specific and specialized connection with a neighboring neuron. It's a synapse, okay? And because neurons are actually not directly connected by a continuous physical chain, but that there is actually a so-called extracellular space between neurons, we need a mechanism by which the information that was sent along the axon here can pass across that extracellular space and be transferred onto the receiving neuron, okay? And that system that the brain developed is called chemical conduction, and it is mediated by neurotransmitters, which are simply molecules which are released by the sending neuron, and they can bind specifically to so-called receptors. It's a bit like putting a key into a lock, okay? So you've got to imagine that the neurotransmitter would be the key and the receptor is the lock. So you just can't put any key into any lock, okay? So just this, in the same manner, you've got neurotransmitters that can bind specifically to a certain type of receptor. And once this coupling between the neurotransmitter and the receptor takes place, then signal can be transferred to the receiving neuron, and it can be strengthened and modified, or it can even be inhibited depending on the receptor type which has been activated, okay? Now, receptors are simply proteins or protein complexes, okay? So they can be mapped using several complementary methods. Here you see positron emission tomography. It's again a non-invasive imaging technique. That's one of the strengths. So it can be used in vivo. The problem is that due to ethical constraints, there's a limitation to how often you can do a positron emission tomography with a single subject because it requires the use of radioactive tracers. And the other problem is also that the resolution that you get is pretty small. You only get several millimeters, so you can't actually see cortical layers, okay? Now, on the other side of the spatial scale, you would have immunohistochemistry, which, if it's coupled with electron microscopy, would let you move down into the range of less than 1 micrometer, okay? That's a big advantage, but it has the disadvantage that you can't detect receptors in their native configuration, right? And in between these two methods, you'd have in vitro receptor autoradiography, which has the advantage of a pretty high spatial resolution, so 20 micrometers. You can see different cortical layers, you can see small subcortical nuclei, no problem at all. And it also has the advantage that you can visualize the receptors in their native configuration, and you can quantify this data, so it allows for direct comparisons not only across brain regions but also across brains from different individuals.
Thanks, Nicola. So which methods did you actually use to measure these receptor distributions in the brain and used in MEBRAINS?
Well, the data that's in MEBRAINS we acquired by means of this quantitative in vitro receptor autoradiography method, okay, the one that's shown in the center of the slide. And the kind of data that we bring into MEBRAINS is stuff like the images that you can see here. These are coronal sections through a macaque hemisphere. These are so-called receptor autoradiographs showing the distribution of 12 different receptor types from different neurotransmitter systems, okay? And the color coding is such that cold tones code for low receptor densities, and warm colors code for high receptor densities. And you can see that the receptors are heterogeneously distributed throughout the brain.
Very beautiful and very colorful images, almost art, Nicola. But what can we actually learn from them? How do you quantify it? And how do you use this type of information?
Well, yeah, that's a really They are very pretty images, and yes, it does almost look like art, Wim. So these images enable a densitometric analysis. So you're basically measuring differences in gray values. Okay, and I'm now zooming in to two of these sections so that you can see a bit more detail, and you can see the different kinds of information that receptors provide. Now the differences in the distribution pattern, this heterogeneous distribution of receptors throughout the brain, reveals different things. So on the one hand, you can see borders between cortical areas and subcortical nuclei. So if you look, for example, at the position here on the left section where these two white arrows are, they are pointing at the position in which this very high density of the muscarinic receptor, indicated by the red colors, decreases very suddenly, significantly, so that you only have, a bit of green and mainly blue colors, okay? And this red part of the brain, we know from electrophysiology studies and from its location here, that is the so-called primary auditory cortex. And you see, if you compare the position of the white arrows on the left section with the position of the arrows on the right side of the section, you will see that for the kainate receptor, we also have changes in density at the position marked by the tip of the arrows, okay? Only for the kainate receptor, we've got the opposite situation to that I just described for the M2 receptor. Here, the primary auditory cortex has a significantly lower density of this receptor type than surrounding areas. And these white lines that you can see here on the left section, they indicate the boundaries of different nuclei that we can find within the thalamus. Okay, so that's one of the things that the differential distribution of receptors show. The other thing is you can see differences between cortical layers. So if you concentrate on this position here, this is the posterior wall of the central sulcus. You will see that for the M2 receptors, the higher densities are found mainly in the superficial layers, the top half of the cortex. You've got red down here because and in the deeper layers, everything is just more like blue, whereas in the kainate receptors, you've got the opposite situation. The deeper layers show higher densities than the superficial layers, okay? And these differences in laminar densities are important because we also know that these layers are the origin and the target of projections in the brain, so where the axons depart from and where they reach. Now, the other thing that differences in receptor densities tell us about are also cortical types. Cortical types means was this part of the brain already present in the brain of very primitive animals, or then we would talk about archicortex or mesocortex, or is it only present in more in species that are more closely related to humans? Then we would talk about a type of cortex that's called neocortex. And when you look at animals, more primitive animals, the proportion of archicortex, which here is highlighted only by this very little piece of a green line here, or the mesocortex, which would be this red part here and this red part up here, that in proportion to all of this black line, which is the neocortex, the neocortex becomes proportionately bigger in humans than in primitive animals, okay? And another thing that we can look at also and analyze is differences in functional systems. So this part of the brain here, we know from physiological studies is involved in the processing of sensibility, so touch, differences in temperature, whereas this part of the brain here is involved in motor control. And you see they have, completely different receptor architecture. Right. And the other important thing about this method and how we can integrate this data into MEBRAINS is, remember that I told you earlier on, Wim, that this method has the advantage of being a quantitative method. Well, what we can do is define regions of interest, and these are simply these little squares that I've defined here for areas TE1 and 3B and 4P, okay? And we can extract all of the pixels that are located in these regions of interest, and we can quantify their receptor densities and express them in numerical form, right? And for every single receptor type that we analyze, we can extract this kind of receptor density, and we can do that in a unit as precise as femtomoles per milligram protein, total protein in the brain, okay? And this is really a very, very small unit. You must think that this would be comparable as to being able to detect a single grain of sugar in a whole mug of coffee, okay? So what we did was extract this kind of data here for very, very many areas throughout the macaque brain. And then we performed with that data a so-called principal component analysis. So each one of these names here represents a single area of the macaque monkey brain. And for each one of these areas, we have this kind of information here. Okay, so all of that is integrated to create this plot here. And if you look at the areas that are located in this plot, and you look at the position in the brain, and you compare not just their location in the brain, but also what their neuronal densities are, you can see that the areas that are located on the left of the plot, they're characterized by a relatively low receptor density per neuronal density, whereas the areas that are located to the right of this first principal component analysis have a very high density of receptors per neuron. Okay? This is because when we move from the left to the right of the principal component, we find more receptors per neuron, because these neurons to the right are also characterized by having larger dendritic trees. And we know that the receptors are normally located on the dendrites. And more specifically, they're located also on the spines of dendrites, and the neurons located in these areas here have more spines than the neurons that are found in these areas down here. And if we then look at what are the differences between areas that are located on either side of principal component number 2, then we will see that basically the difference between these areas up here and the ones down here is the relative density of the 5-HT1A receptor, which is very high in these areas up here compared to the areas that are located down at the bottom of the PC2. So interestingly, these two axes of the principal component analysis separate brain areas on the one hand based on whether they're associated with sensory functions or if they're more association multimodal areas, and it also separates them depending on whether they're evolutionary origin, phylogenetic origin, if it is older or if they're newer. Okay, so these are the kind of things that you can see. Now, this is an abstract view, okay? If we project the PC1 values onto the surface of the macaque monkey brain and we put each value onto the place in the brain where it belongs, we obtain this pattern here. And this is something that can already be integrated into the MEBRAINS Atlas, okay? Because this is the surface of the MEBRAINS template. So, but this isn't just important just because it's in the Atlas. It's also, if you compare the location of these blue tones versus the red tones in the macaque monkey brain, and you compare them to this image on the right of the slide, which shows you a drawing of the human brain, and which has been color-coded to highlight the location of the so-called human functional zones, you will see a very high similarity, indicating that that the macaque, or not indicating, but confirming that the macaque monkey brain is a valuable tool, a valuable model for the human brain, and also confirming the hypothesis that distributions about receptor transmitters tell us something about the relationship between structure and functional domains.
Very fascinating receptor information, Nicola, at whole brain scale and at very fine spatial resolution as well. But how can this data be related to other types of information? So how do we bring this all together?
Well, Wim, this is a nice question. I'm going to try and get this video to work. Hold on a second.
Oh, sorry.
Come. Oh, no, sorry. No. Now, go back. Right. Well, what we can do while I try and there it is. There. Right. Is it going?
I'm gonna remove the pointer and then I think it will.
Yeah. Well, anyway, because the video on the left is going now. Now it's going. Sorry, guys. This is, technique sometimes makes our life difficult. Right. Well, in the MEBRAINS multimodal atlas, we don't only provide a parcellation scheme based on the analysis of cell body distributions and myelin patterns and multiple receptor types, but we've also created 3D reconstructions of the receptor autoradiographs. And you can see one of them rotating here on the left video. Okay, so this will be integrated into MEBRAINS. It's not available in MEBRAINS yet, but we're actually in the process of making it all of these different receptor maps available via MEBRAINS, and you can then, because they're in the same space as functional images, you can then overlap these receptor maps with your functional activations, for example. So the right side of the video showed activations in a monkey brain. And you can then, for example, if you're using the parcellation scheme, you can use that to do a cytoarchitectonically informed analysis of your functioning imaging data and ask which part of the brain do you is this particular blue activation or red activation located in, or what is the different receptor architectonic pattern in that part of the brain that could then maybe explain why in activity in one particular part of the brain is faster than in another specific part of the brain, okay, depending on whether it expresses, for example, more ionotropic or more metabotropic receptors. Okay, so this is the kind of thing that you can ask with your data to integrate it. These are things that are actually either implemented in MEBRAINS already or just being implemented sort of like as we speak. But Wim, tell me something. What about the future? What about other things that developments that might be coming up? Is there anything new in the pipeline that we can look forward to, say, in the next, I don't know, 4 to 6 months?
Yeah, well, of course, you can do many things. I think many people are now recording with many electrodes simultaneously from the brain, and then you can bring that information to here. You can relate it to, as we said in the beginning, to written topic maps, distributions of receptors, exactly as you just told in a minute ago, Nicola. But other fascinating thing that we started to do recently in our lab is functional ultrasound, which is an imaging modality, a functional imaging modality, which has a much, much higher resolution than fMRI, with the only caveat that you can only image a restricted portion of the brain, as is here indicated, about 1 cubic centimeter, but at an astonishing resolution. So you really get, and a high sensitivity, you really get very, very very specific information at different layers, and at, let's say 100 to 200 micron resolution, which is like 2 log units better than typical fMRI experiments. So this is information that is also relatively easy to gather, and I suspect that this will sort of like revolutionize imaging in the macaque world. On top of that, this kind of information can be related to human data. We forgot, and we don't have time to go into it, but on the human front in EBRAINS, they're also building these receptor density maps and so on and so on. So I think by bringing these different atlases, not only MEBRAINS, but also the human atlases into play here, that it will become a very interesting playground, that every individual single experiment becomes much more valuable, because it's easy to relate the experiment-specific information with much more information with a click of a mouse away from it. So I think it's going to be very fascinating. It took a while to build it, but I think it's going to be a very fascinating era to come. I think we are nearing the end of our webinar, Nicola. And obviously, I also like to emphasize that by no means it is just you and me that did this.
No.
Actually, basically, it was done by a bunch of other people. Some of them already left, and some of them are still very active in sort of like improving the atlas, and also to populating the atlas, which I think is very important, of course. This is a very daunting task. Also, the annotation team and the curation team from EBRAINS is exceedingly helpful and needed to actually get the data in a fair, possible manner implemented in this data. So I would like to thank all the collaborators here that put a lot of effort and time in making this available. And obviously, I also would like to thank the audience for listening to us for about an hour. And obviously, stay tuned. So here you can follow up what will be next. And we are also happy to address some questions if there are questions from the audience. Thank you very much.
Yeah, thank you. And the other thing also is, Wim, that you and I, we're not the only ones that are allowed to put data into
Yeah.
MEBRAINS. Anybody working in the macaque monkey brain who would like to, add any kind of data to this is more than welcome, and they just have to contact the EBRAINS curation team, and they will then be pointed in the right direction.